through mitosis, produce a gamete, and then that gamete gets fertilized, right? Becomes a zygote, and then it goes through meiosis to produce the haploid individuals that just perpetually live. And plants are able to split this alternation of generations, at least some plants are, 50-50. Half of their life is spent as a haploid organism, and half of it is spent as a diploid organism. So our sexual life cycle looks something like this. We have adults that go through meiosis to produce gametes, either an egg cell or a sperm cell. And then there is fertilization that occurs to create that zygote, right? Meiosis produces the gametes, and then mitosis produces a multicellular organism from that unicellular zygote. And then after this stage, it's all mitosis to get back to that adult and sexually mature stage. Okay. So I'm really quickly going to introduce meiosis 1 and talk primarily about the differences between meiosis 1 and mitosis. So the first stage, I'll keep a list on this side.
Someone tell them this. There's someone sneezing.
Is everything okay? Okay, we're good. Okay, so the first stage of meiosis is meiosis 1. And meiosis 1 consists of 4 stages. And remember, we're going to call these air quotes distinct stages, right? Because it's just one long process. It's one continuous process. Prophase 1 in meiosis 1 has all the same things that happen in prophase of mitosis. So the nuclear envelope begins to dissolve, chromosomes begin to condense, and the mitotic spindle begins to grow. Importantly, however, homologous chromosomes in prophase 1, and this is the difference, homologous chromosomes begin to pair up. It's a process called synapsis. So in prophase 1, we get what's called synapsis. And synapsis is homologous chromosomes pairing. So that would look something like this, where we have our cell. We have our nucleus that's beginning to dissolve. So I'll just kind of draw a dashed circle. And then we have homologous chromosomes. Now, please remember from our discussion of mitosis that homologous chromosomes are 2 chromosomes that have the same genes on them. One you get from mom, one you get from dad. So when homologous chromosomes pair in meiosis, in prophase 1, then that chromosome that we got from mom, let's say, that carries a gene for like eye color, pairs with that same gene, that same chromosome of dad's, that also codes for eye color. And by pairing, it's like they're just lining up right by each other. They're just right next to each other. And you get that for all of your homologous pairs. So for us, it would be 23 pairs of homologous chromosomes that are all pairing up. Yeah, so I drew them kind of wiggly because they haven't completely condensed yet. Now this stage is really, really important because another phenomenon happens during synapsis, and that's called crossing over. In crossing over, what happens is 2 homologous chromosomes will exchange some DNA. So like the leg of dad's chromosome will flop over on the leg of mom's chromosomes, and they'll actually break, and what was part of dad's chromosome will become part of mom's, and what was part of mom's will become part of dad's. So in crossing over, chromosomes, homologous chromosomes, and I'm just going to write H period for homologous chromosomes, exchange material, exchange DNA. We used to think this was a really uncommon thing, but now we know that each homologous pair will go through crossing over like 3 times for each division. It's really, really common. And that's a really important way to introduce genetic variability. Ultimately, and I'll talk about this a little bit later when we talk more specifically about crossing over, ultimately what it produces is a recombinant chromosome. Okay, so that's prophase 1. All the same stuff happens in prophase 1 that happens in prophase of mitosis, right? We get chromosomes condensing, nucleus falling apart, blah, 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 blah. Mitotic spindle growing out. But the difference is, in prophase 1, we get pairs of homologous chromosomes that are lining up together. We get that pairing. That pairing is called synapsis. And synapsis facilitates crossing over. Okay. Now, homologous chromosomes are held together by those same proteins that hold sister chromatids together. Do you remember what those proteins are called? The centromeres are the things in the middle. Yep. It's those cohesins, right? It's the cohesin proteins that act like a zipper. Okay, so in metaphase 1, remember in metaphase of mitosis, all of the chromosomes were lined up at the center of the cell. In metaphase 1, we get pairs of chromosomes that are lined up at the center of the cell. I'm just going to draw a picture of that instead of writing it out. So we have our cell, and I didn't draw the spindle. In metaphase 1, we have our pairs of chromosomes that are completely condensed now lined up at the middle of the cell. So we have mom's chromosomes and dad's chromosomes. And our spindle is grown out like that. But we also have those other weird microtubules that are growing out and attaching to each other, right? Do you see how that's different than metaphase of mitosis?
The microtubules are.
So, and that'll be similar to metaphase and mitosis. All the microtubules kind of look like that, but the chromosome orientation is quite different, right?
Yeah.
So if this was metaphase in mitosis, all 4 of those chromosomes would be lined up on top of each other. So it would be like blue, blue, orange, orange, all on top or whatever. Okay? So what happened in anaphase of mitosis? What's that? Yeah, and what starts separating? Chromosomes. Sister chromatids get pulled to opposite ends of the cell. And so with this orientation, what do you think is going to be pulled to opposite ends of the cell? Yeah, like the whole chromosomes, right? So you're going to get 2 chromosomes going this way and 2 chromosomes going that way. Does that make sense? Mm-hmm. So, difference number 1 in prophase 1, synapsis, right? And we also get crossing over. Difference number 2, in metaphase 1, we get homologous pairs aligned at the center. Difference number 3 in anaphase 1 is that chromosomes are pulled to opposite ends of the cell.
So could that be really bad if it pulls like the whole thing over?
For sure. In Sarai, that's exactly how trisomy 21 occurs. It generally occurs in the production of an egg, and that egg doesn't separate at metaphase 1 correctly. Or metaphase 2. We'll talk about that a little bit later. I'm out of time. Yep, go ahead.
What was the question for the or what was the difference for the anaphase one?
Anaphase 1 is homologous chromosomes are separated and not sister chromatids. So a whole chromosome is being pulled the opposite end of the cell. But we'll pick up with anaphase 1 when we come back after Thanksgiving. Okay, you guys have a great and safe Thanksgiving.